Alright, so today we're going to look at the last section of this unit. It is mutations, how things change or can change and what the effects are. Right, which can be pretty severe depending on what happens. Right. So mutations. A mutation is a change in the DNA sequence. Right. We're looking at simple gene mutations right now. Alright, we have two types. We have point mutations, which pretty much tell you what it says. They happen at a single point. Alright. And then we have frame shift mutations. Alright, two different types. Mutations, the word itself comes from mutagen, caused by a mutagen. A mutagen is something that causes some things to happen, things to change. All right? So it could be chemicals, it could be radiation. Heck, even smoking can change your genes. And one of the bad things about smoking. Okay, so if we look at this example here, we see a strand of normal DNA at the top, and then we see a mutated strand. That mutated strand, it's simply there's one base difference. Could be a problem. Might not be a problem. All right, we'll see how that works here in just a second. All right, point mutations. Point mutations are something that happens with one single nitrogen base at one point on the DNA. Okay, that's where that word point mutation comes from. It happens at one single point on the DNA. All right. We have three types. We have silent. And when you're silent, no words come out of your mouth. So what this means is when it's silent, nothing happens. Okay? So that base change occurred, but when it occurred, it's still coded for the same amino acid when the mRNA was decoded by the ribosome. And we'll look at that down here in just a second. The second one is nonsense. All right? Nonsense means that one base changed that amino acid from whatever it was to a stop, and the decoding process stopped there, so the rest of the gene did not get decoded. All right. Missense means it codes for a totally different amino acid, and that one single amino acid is changed, that 1,3 base sequence. Okay. So here's a normal DNA strand. And what would happen during transcription translation. Okay, so we have that normal strand. It's supposed to code for tyrosine. Now, a point mutation happens here where that original G is turned into an A. It was coded or made wrong. Now, you see that this normal mRNA is not the same anymore. It codes for UAU now. But UAU and UAC both code for tyrosine. So this right here would be a silent mutation because everything is still the same. Okay? Nothing changed. You wouldn't even know you had this mutation. The next one shows that that G was changed to a C. Well, that C is going to then change to a G when we go through transcription translation, and that codes for a stop. All right. Yeah, we've changed it now. Everything after this point doesn't happen because the ribosome sees stop. This is a nonsense mutation. All right. It means that it changes from that point on. Okay. In the last one here, the first base was changed from an A to a G. Changing that gives us the amino acid histidine. It's not tyrosine. It's something totally different. This is a missense mutation. That gene's not going to work right. The protein's not going to be built right. Not going to see what you're supposed to see. Okay. The second part are frame shift mutations. Frame shift mutations come in two forms, insertion, deletion, and it's just what the word says. For insertion, it means that something is being added. For deletion, it means something is being taken out. Okay? So we give this example here. Here's our example. Notice 3 letters, 3 bases. The dog bit the cat. All right. But if we look at this second one here, something happened. This O, that base, is gone. It's not there anymore. All right. And where the word frame shift comes in is that, remember, the ribosome is going to read 3 bases at a time. It doesn't know that that one's missing. Okay? It's not going to just leave a hole there. It's going to read 3, and then 1, 2, 3, which changes this amino acid now from DOG to DGB. Well, at the same time, it changes the next one, all the way down. It shifts the entire reading frame that way by 1 base. All right, we deleted 1 base, and now everything has changed from that point on. Okay. Oops, sorry. Adding a base would do the same thing. If we were to add another O in here, it was DOOG. All right. So the second one would not read DOG anymore. It would read D. My pen is not working. O, and then it would read G, B, I, and so on down the line. It shifts the reading frame the other direction by adding a base. All right. Insertion. It would shift it the other direction. All right, so we have our mutations here. This one is insertion. Here's our original strand, cat, 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 cat. All right. But then on the mutated strand, we see we have an extra A in here. It's been added. And notice it shifted the entire reading frame. Because now that cat that used to be here, there's the original, is not there anymore. We have ACA, TCA, TCA. It's going to change everything after that point. All right. That protein is not right. It's coded wrong. It's not going to work, which means this gene is not going to be expressed properly. Okay. Now, are all mutations bad, harmful? No, they're not. Silent mutations, you'll have these. You might have these in you right now, and you don't even know it, because the amino acid still, or the 3-base sequence still codes for the same amino acid. Doesn't hurt you. All right. Mutations can help you, all right, and can help other organisms if, and only if, they lead to a trait that helps the organism survive. All right. For instance, if you have a black organism, say a black rabbit that lives in a snow-covered environment, he's not going to survive. He stands out. He's going to get picked off by predators. But if something happens in his genes that they mutate, they change, and now all of a sudden his fur is white, it helps him in that environment. So all his offspring then, you're going to start to see white rabbits, because the black rabbits are going to be picked off by the predators. The white rabbits are going to survive. You're going to see that change in the color of organism. We're going to talk more about that next semester when we get to evolution. Okay. So that's mutations for now, the gene mutations. In the next unit, we'll talk more about mutation. We'll talk about chromosomal mutations. But these are just gene mutations. One simple thing happening inside that DNA to allow something new or something different. Both can be harmful and helpful.